Hello and welcome to GED Live at Phuket PALS with teacher Alex to another GED science screencast, this time on chromosomes, DNA and the genetic code. Alright, let's get started. So in this presentation we will have a look at chromosomes and gender, DNA, the molecule of life, and DNA and the genetic code. We will start with the chromosomes and gender. Inherited characteristics. What makes you human? What determines your gender? In all living things, characteristics are passed on in the chromosomes that offspring inherit from their parents. So all human characteristics, including gender, must be something to do with chromosomes. And where are chromosomes found in cells? Chromosomes are long strands of genetic information located in the nucleus of a cell. They are made of a long molecule of DNA wrapped around so-called histone proteins. Chromosomes are most visible during cell division, when they are replicated and packed up, condensed, and look like, well, this X-shaped form. I'm sure you've seen that before. Homologous chromosomes. In most cells, chromosomes are matched in pairs based on their size and shape. Matching pairs of chromosomes are called homologous chromosomes. Homo means same, logos structure. So homologous structures means, or homologous chromosomes means we have two chromosomes with the same structure. Where do these homologous chromosomes come from? One of them comes from our mother, from the female parent, and the other comes from our father, our male parent. So each pair of homologous chromosomes contains one chromosome that has been passed on, inherited from each parent. One comes from the mother, and the other comes from the father. We have learned in a previous presentation that we will see this here again later, that we have 46 chromosomes in total in our cells, and we get 23 of these from our mother and the other 23 from our father, so we have 23 pairs of chromosomes. So a quick word to chromosomes themselves. Chromosomes can be a bit confusing in terms of their the vocabulary. When do we use the word? And because there are different types of chromosomes, basically, depending on what state the cell is in. When the cell has just divided and we have a fresh cell, a new cell, then the chromosome looks like this. It's one leg, and this chromosome is usually not visible like this in the cell, because this is packed up DNA. The DNA will be spread out, uncoiled, and so the cell can use the DNA and make proteins. When a cell is about to divide and split into two new cells, then both new cells need a complete set of chromosomes. So the chromosomes that look like this, they need to replicate, they need to double the genetic material. And then they look like this. This is one chromosome and this is another chromosome, and each of these chromosomes has two sister chromatids, which are exact identical copies of each other. And they can then be split in the middle, and one of the halves goes to each cell that will form new. As we said earlier, human chromosomes. In the human body cells, there are a total of 46 chromosomes in each cell. How many pairs of homologous chromosomes are there in a human body cell? 23 pairs. You inherit half of your chromosomes from your mother and the other half from your father, so 23 of each parent. How many chromosomes do you inherit from each parent? 23 unpaired chromosomes. They come together and we have 23 pairs. The X and the Y chromosomes. special pair of chromosomes. These are the sex chromosomes. Two types, the X chromosome and the Y chromosome, the X chromosome being much bigger than the Y chromosome. And what makes a girl or a female is having two X chromosomes. And she gets these from each of her parent, one X chromosome from the mother and the other X chromosome from her father. Males, on the other hand, have one X and one Y chromosome. The X they inherit from their mother and the Y from their father. So the chromosome pairs of sex chromosomes in a female look like this, two X chromosomes, and in a male look like this, X and Y. If the male makes sperm cells, we have a 50-50% chance that a sperm cell gets either the X chromosome or the Y chromosome. So the sperm cells will determine the sex of the baby. Here we have an example of a female human karyotype, which has two X chromosomes. As we can see, the last chromosome pair, sex chromosomes, 2X. The homologous chromosomes, as we already said, they have the same size and they have similar banding, so similar genes basically. And we can see that very well here, the orange and the green color representing the banding of chromosomes. What is active DNA, inactive DNA, what are genes, what are not genes. And we see that every chromosome has its homologous counterpart, his homologous partner. 23 pairs of chromosomes. A male karyotype would look different only in the last pair. We would have one X and one Y. Chromosomes and sex cells. So what happens to the sex chromosomes when sex cells are formed? Here we have a female body cell which will undergo meiosis, so-called reduction division, to form egg cells. Again, our sex cells, eggs and sperm cells, they need half the number of chromosomes because when a new human is made, a baby is made, an egg cell and a sperm cell come together, they fuse together. So we have 23 chromosomes and 23 chromosomes adding up. That means before we need to reduce the number of chromosomes in the egg cells and the sperm cells. So the egg cells are made from female body cells, and we can see that the X chromosomes will split up. So each X cell has an X chromosome. Here we have a male body cell that will undergo meiosis to form sperm cells, and we can see we have a 50-50 chance of having either an X or a Y in the sperm cell. So why is there an equal chance of having a boy or a girl? 50-50% chance, because 50% of the sperm cells have an X chromosome and 50% have a Y chromosome. Alright, let's go to our next part of the presentation, the DNA molecule of life. So what are chromosomes made of? Chromosomes carry the genetic information for making all living things, everything from a human to a microbe. Why are chromosomes known as instructions of life? And what are these instructions of life made of? So here we see that chromosomes are actually made of DNA. And every one of you heard of DNA before, and we will now explore what this DNA actually is. Chromosomes and their genes are made of a molecule called DNA. DNA molecules carry the code that controls what your cells are made of and what they do. DNA stands for deoxyribonucleic acid, and chromosomes are tightly coiled and packed up molecules of DNA that are wrapped around proteins and then wrapped up again and again and again until we get a supercoiled structure called a chromosome. Which part of a DNA molecule holds this information? So let's have a quick look at the structure of DNA. DNA is, first of all, a double helix, which means a DNA molecule consists of two strands that wind around each other like a twisted ladder. Our famous DNA double helix, alpha helix. The DNA has a backbone the portion of the DNA double helix that provides structural support to the molecule, and each strand of the two strands has its backbone made of alternating sugar and phosphate groups. The sugar is called a deoxyribose, which we can see here as an S, and phosphate P. So sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. That's the backbone of the DNA. Both strands, this is going to be the same. So, what else do we have in the DNA? We have this bit in the middle here. These are the so-called DNA bases, or the base pairs. The rugs of the ladder are made from these pairs of bases. There are 4 types of bases: adenine, guanine, thymine, and cytosine, and they are usually assigned the 4 letters A, C, G, T, so we don't really need to remember these names. Important is to know the 4 letters A, C, G, and T. And these bases always pair together in the same way. Well, if you have a look at these pictures here, how do you think the base pairs will pair up? You actually see that here already. G pairs with C and T pairs with A. As we can see, they match perfectly. The base pairs hold the 2 strands of the DNA double helix together with relatively weak bonds, so-called hydrogen bonds. You can see here, a bit difficult to see, but we have these dotted structures, which are weak hydrogen bonds. And in the sum of these thousands of base pairs, that gives our double helix quite a lot of stability. There are millions of base pairs in a DNA molecule, and that always follow the same rules of pairing. The rules of pairing are A always pairs with T, and C always pairs with G. And amazingly, it is the sequence of bases along a DNA molecule that forms the genetic code. It's that simple. It's about what comes after one base and the next, and so on. So here we could have the sequence of the genetic code being C, A, G, T. This is how the genetic code works, and we will explore that in the next part of our presentation, DNA and the genetic code. So why are cells different from each other? An oak tree, for example, has about 30 different cells, types of cells. How many different types of cells does a human have? Our bodies have more than 300 different types of cells. Why do cells have different characteristics? Different types of cells produce different types of proteins. For example, keratin is a protein in hair and nails and some skin cells. Outer shells of insects contain hardening proteins called chitin. It makes the hard skin of insects. Enzymes in plants control photosynthesis. Enzymes in plants and animals control cellular respiration. Enzymes in general control the chemical reactions that happen inside a cell. Enzymes are proteins. Elastin and collagen are other proteins in the skin that give it flexibility and strength. So we have many different types of proteins, and these different types of proteins are expressed in some cells more than in others. And all of this is controlled by the genes in the DNA, by the genetic code. So what's a protein made of? Let's have a look at this first. Proteins are long molecules made from chemical units called amino acids. There are 20 different amino acids, and we have basically unlimited ways of combining them. Special combinations and folding will result in a specific protein with a specific function. So here we see a part of a protein molecule and the sequence of amino acids. What happens if amino acids are combined in a different order? Well, then we get a different protein. Very important to mention here that not any combination will result in a functioning protein. So we need a specific combination for which the information is stored in the DNA, and that specific order will result in a functioning protein. So let's connect proteins and genes. Consider the two following statements. Genes carry the instructions for inherited characteristics. Cells have different characteristics because they make different types of proteins. What is the connection between genes and proteins? Genes contain the instructions for making proteins. What molecule has the instructions for making proteins? Exactly, the DNA. Alright, how do genes make proteins? Genes are made of DNA. Genes are part of a chromosome. A certain length of DNA can be a gene. And proteins are made from amino acids. And each amino acid is coded for by its own special sequence of 3 bases. And these are called triplets or codons. So 3 letters of our DNA, 3 specific letters, will equal one of our 20 amino acids. So, how do genes make proteins? The order of triplets in a gene determines the sequence of amino acids. The amino acids join together to form a protein molecule, and each gene contains the sequence of bases for one protein. As we can see here. Here we have a part of a gene. We have the triplets, and each triplet, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, will code for a specific amino acid. Joining them together, we get our protein. This translation from the genetic code and the triplets to our connected amino acids, this is done at the ribosomes. Ribosomes are special structures inside the cell that have exactly that function, building proteins from the information in the genetic code. For more detail, you can watch the presentation on cell structures in animal and plant cells. So why is the sequence of bases in a DNA called the genetic code? How do genes make proteins? The genetic code is the order of DNA bases which determines the sequence of amino acids in a protein. How many triplets code for a protein of 20 amino acids? So 1 amino acid is equal to 1 triplet. So 20 amino acids equals 20 triplets. How many bases code for a protein of 20 amino acids? 20 times 3, 60. So, let's try ourselves. Here we have different triplets, and each triplet equals an amino acid. Now we have a gene down here, and let's try if we can translate that gene and do the ribosomes job. Our first triplet is green, yellow, red. Where do we have that? Here, exactly. So we take this amino acid. Our next triplet is red, red, red. So we take this amino acid, connect it to the other one. Our next triplet is pink, red, pink. So we take this amino acid and connect it. Our next one is pink, pink, green. So we take the triangle, another amino acid, connect it. Our next one is yellow, red, green. Yellow, red, green is here. So we take this amino acid and connect it to our growing protein. And our last one is this amino acid again. We connect it, and we have our finished protein. This is exactly the job that a ribosome does. It reads triplet by triplet and grabs the correct amino acid, connects it to the growing protein chain until the complete gene is translated and we have our finished protein. So how does evolution happen, and how do new proteins come into existence? Again, we've gone a long way from unicellular organisms like bacteria to complex multicellular organisms like many animals are. New proteins may come into existence by changes in the DNA that can happen randomly. These changes are called mutations. A mutation is a change in the sequence of bases in the DNA. Mutations can be caused by mistakes in copying the DNA or the effects of radiation or heavy metal ions. Will the mutated version of DNA make the same protein? No, it will not, because this triplet has changed here compared to that. So in this case, one amino acid will change. Most of the times these mutations will likely have a devastating effect for the protein that it just won't function anymore. Very rarely, a mutation can lead to a protein that has a new function, and eventually that's a beneficial function, giving an organism an advantage over its partners in the same species. And that slowly could lead to the new protein being found in more and more organisms of the species, and eventually lead to a change in the species having a new protein, a new gene. A DNA mutation changes the amino acid sequence, and so a different protein may be produced, as we just said. If genes produce incorrect proteins, cells may not function properly. This is the cause of many inherited diseases, for example. So inherited diseases are basically mutations caused by mutations in the DNA that lead to a protein not functioning properly. We can see that here our mutation was in this spot. That leads to an exchange, to a change in the amino acid sequence of the protein. Alright, that's it from me with GED science on chromosomes, DNA and the genetic code. I hope I could help you with your preparation for the GED test. If you have any questions or need to know more, please feel free to comment on the video. We will get back to you. Or if you need more focused help on your preparation for your GED exam, please feel free to contact us. Have a great day ahead, and I'll see you next time for another class on GED science.